Okay, good morning, everyone. We're going to start the lecture. So please take your seats. We'll take attendance at the end, because yesterday, when I did the attendance at the beginning, it was actually a lot of disruption and waste of time. So I'd rather waste the time after I finish the lecture. So please take your seat. You should start on time. You're going to be professionals. You actually should respect that you should be on time, and you actually need to respect the people as well. So today, we have the second lecture on the histology of the digestive system. We'll cover the esophagus and stomach. Please stop talking and listen to me. If you are not interested, you can just leave. And somebody close the doors in the back if you can. Okay, so today's lecture is about the esophagus and stomach, and Dr. Akif just covered the gross anatomy of these two organs. So today we're going to talk about histology. So the objectives of the lectures today, we will describe the layers of the wall of the digestive tract, the general overall structure of the digestive tract. We'll also describe the structure of the esophagus, the location and function of the major nerve plexus in the esophagus. We'll also describe the gastric glands. We'll list the types of epithelial cells that's present in the stomach and the major function of each. And we'll try to have some understanding of the many steps of acid production by the parietal cells in the stomach. Yesterday, I showed you this slide, which was the overall structure of the digestive system, and actually have glands, tract, and accessories. Yesterday, we talked about the tongue, one of the accessories, and the salivary gland. Today, we start talking about the tracts, starting with the esophagus, and followed by the stomach. So first, you remember yesterday, I told you about with histology, there are patterns you should follow. A lot of time, we found the arrangement of the layers of the presence of the cells takes certain pattern with different organs or different tissues. Today, especially the GI, there's a very, very particular pattern of the layers. So there is a general structure that we apply to anything from the esophagus all the way to the rectum, the arrangement of the layers. So the first one, or the innermost one, and is facing the surface outside. In this case, because the structure has a lumen, it will be facing the lumen. The first layer is called the mucosa. It's formed by several different layers. The innermost or outermost one, in this case, because it's going to be facing the lumen, it's going to be the lining epithelium. This will vary between the organs. But when you look at the tube, the cross-section, the innermost, or the one that's facing the lumen, is going to be the layer of epithelium. And you remember from the general structure of the epithelium has always a free surface facing outside. This will be underneath, will be followed by another layer, underlying connective tissues called lamina propria. And I think you've been exposed to that before. And in this, the general structure of the GI, there's another layer called the smooth muscle layer called muscularis mucosa, which we haven't seen actually much. But when you look at the mucosal layer, the free lining epithelium underneath, underlying connective tissue, and the smooth muscle. And this actually, those two pictures here, one of those is an illustration showing you the arrangement of the layers, and the other one is the histology. So in the histology, you can see here there's a mucosa. It's difficult to identify the three layers of the mucosa because of just the magnification. But I put this image for you to see the general structure and the arrangement. Under the mucosa is always the submucosa, which is a dense irregular connective tissue, then followed by muscularis. Some people call it muscularis propria in some of the books, but I think now they all agree it's called muscularis, just leave it as is. It's a muscle layer. Two layers of smooth muscle, generally, except we'll talk about some exceptions. And also followed by the fourth layer, which is the serosa, or sometimes called adventitia, based on the organ. So the serosa, if you found a membrane of a mesothelial cells, very thin, squamous cells making a layer similar to what you see in the blood vessel. If it's actually lining a cavity, we call it mesothelial. It's the same, squamous epithelium. If it's making the blood vessel, we call it endothelial. If it is making a cavity, like the peritoneum, we call it mesothelial. And I probably remember, at least from general talks, that mesothelioma is one of the diseases of cancer that arise from this epithelial cells. And you'll find a small amount of connective tissue, we call it adventitia. So the word serosa and adventitia is actually very interchangeable. People use them differently. And sometimes just based on how they are harvested. If you have a serosa and you do not pick it up in the tissue, then you're not going to see it, you just see connective tissue. So these are the arrangement of the layer. It will always be this pattern in the next few lectures when we talk about the GI, esophagus, stomach, intestine, small intestine, large intestine, mucosa, submucosa, muscularis, adventitia, or serosa. So here's an illustration showing the 4 layers, and this is actually how they look under the scope. Mucosa, submucosa, muscularis is thick layer, and then adventitia, you can see a little bit of it. If I'm talking too fast, let me know, because I think I got a note yesterday from the department chair that I talk sometimes fast, which I know I do. So it's not. So again, try to link the structure-function relationship. So what's the function in the esophagus? First, the esophagus is not just simple conduit. It's just a tube. You drop something and goes to the stomach. It actually has more than that. It contracts and in a synchronized manner to cause this peristalsis move. So to move, your food goes down, actually push the food behind and then relax in front. So to keep moving. If actually it contract all the time, it actually causes severe pain. And actually this is some of the problem with some diseases. It causes a similar pain almost like an angina or heart attack. Extremely painful condition. And while the muscles behind the foot contract, the one, as I said, in front relax. I don't know what the funk is. I think I need to fix this. For some reason, I think it went beyond the boundary of the slide. So the first organ today is the esophagus. Again, remember the 4 layers we talked about. We'll start talking about the mucosa of the esophagus. Again, this will be the innermost or outermost based on how you see things. If you're looking at the tube, the inside, if this is the innermost, if it's facing outside, it's outermost. So anyway, it's the one that's facing the lumen. It is made of stratified squamous epithelium, it's non-keratinized. Provide protection from abrasive food. You remember the skin also is stratified squamous, some keratinization, but the skin is for protection. It's similar with the esophagus. You can't imagine how much stuff we shovel in. Very abrasive. I mean, those pretzels and potato chips, they're very abrasive. They can cut a thin mucosa. So luckily, we have this stratified squamous epithelium can take this kind of thing. In actually some animals, you'll find this keratinized. If you get a sample from a dog or something, because they eat hard food, you will find this will be keratinized. Just again, because of the structure-function relationship, they eat harder food than we do. We don't have anything, nobody cooks for them, so they actually have to eat anything they can. Followed by the lamina propria, simple loose connective tissue. Always in the lamina propria, you'll find those diffuse lymphatic tissue. In the esophagus, they have what's called cardiac glands, and we'll talk about that in details. And then muscularis mucosa. This is longitudinally organized smooth muscle, which is thick in the proximal part of the esophagus. It helps with the swallowing. This is actually, if you look at the image here, we have the stratified squamous epithelium, the lamina propria. You can see all these little cells are inflammatory cells or immune cells, lymphocytes, and other eosinophils and basophils. And this is the muscularis mucosa, smooth muscle layer. So if you look at this specimen, it's going to be always a cross-section usually in the GI tract. So if it's longitudinally arranged, it actually will be like a cross-section in your section when you look at it. If you imagine this is the longitudinal section, the muscle running like this, when we expose, we cut the section like this, it will be like a cross-section. So if somebody asks why it sits longitudinally, but we see it's cross-section, just because of the orientation. It doesn't mean the muscle changes its position. In the submucosa, it's a dense, irregular connective tissue, a lot of collagen. It also, as we said, some of the general structures of this, it always harbors the lymphatic vessels, nerve fibers, ganglion, and other things. There is a particular name for the nerve supply in this area. It's called the Meissner plexus. And we'll talk about some of those plexuses also later in the lecture. There's always diffuse lymphatic tissue and lymphatic nodules. So lymphatic tissue, which you get lecture on, I think next 2 blocks or something, is always there because it's part of your immune system. So you have to have those somewhere. Sometimes they're actually scattered cells, but also sometimes they form these small nodules. They'll be around its structure, and sometimes you can spot them. And I remember some of the students, they asked me in the lab sometimes what it is, yeah, because you just didn't cover it here. But yeah, you see all these lymphoid structures scattered everywhere in the mucosa and submucosa. The esophageal gland proper, if you look at the submucosa, there's actually certain glands here similar to, it's part of the salivary system. We mentioned yesterday there's a gland that's called the major one and the minor one. This is considered one or the minor one. They get also some lubrication to the surface of the esophagus. You need the esophagus to be wet, not to be dry when you eat the food. So they are scattered along the esophagus, more concentrated in the upper half. And they are compound tubular-alveolar gland. You remember yesterday I told you the structure, the general structure of the gland. It can be tubular, it can be long, it can be alveolar or acinar rounded, it can be combined, it can be branched, it can be simple. So these are compound tubular-alveolar. So they're actually, if you look at the image that I gave you yesterday, they're actually multi-branches, and they actually mix both tubular and acinar or alveolar. You should be able to recognize them easily. Sometimes you can see actually big aggregates. Sometimes in the section you can see less. This is a special stain, so they look actually clear. Sometimes it's going to be like more of a mucinous serous kind of structure. The excretory duct, it's usually stratified squamous epithelium, again similar to what you can see in what you can see in the salivary gland yesterday. So here you can see an image of a duct. You can see how big it is. This is stratified squamous. And again, consider part of the salivary gland of the oropharynx. And this gland secrete mucus. It's usually slightly acidic, but lubricate the lower part of the esophagus and protect this from acidic stomach secretion. We'll talk about this GERD, which is the disease we're going to talk about, gastroesophageal reflux disease later. The third layer is the vascularis. Again, inner circular, outer longitudinal. They'll always be this array. The inner one is going to be circular around the tube. The outer will be longitudinal. Again, when in the cross section, it's going to be the opposite to you when you look at them. Again, the type of muscle here, again, this is part where it's actually unique. It's mixed skeletal and smooth muscle. The upper is made of striated muscle. The lower is smooth. The middle one is mixed. Again, the 1/3 is a rough estimate. It's not really 1/3. The people said in the lecture, at least in the literature, they said at least 50%, which is the lower part, not really 1/3. But just for simplification, the books put it 1/3 striated, 1/3 lower smooth, 1/3 mixed. Because remember, you have to actually take a voluntary action to swallow. You know, the food, you don't put the food in your mouth and then it automatically will drop in. You actually have to swallow. So the striated muscle initiate, but after that, in the middle, you don't need to. Once you decided to swallow, it's actually going to keep going. You know, there is no one is going to change in the middle of either the bolus in the middle. Now I'm the one that's, you know, swallowed anymore. So there is no reason for this. So again, the function of this to help you to start the swallowing process. There is another plexus, which is called the Auerbach plexus or myenteric plexus, present between the two layers of the muscle, between the outer and inner layers of the muscle. There's this Auerbach plexus. It's part of the enteric system, and we're going to talk about that later, you know, in a different slide. So the striated muscle in the upper part is innervated by the somatic motor neuron of the vagus nerve. The smooth muscle of the lower part innervated by the visceral motor of the neuron of the vagus nerve. Again, the fourth layer, which is the adventitia, again, depends on the organ. Sometimes it will be the serosa, sometimes adventitia, just outer layer of the esophagus. It's connective tissue, basically, and harbors some nerves and sometimes, you know, lymphatics. To summarize this part, we cover the general structure of the GI tract and the 4 layers, the mucosa, submucosa, muscularis, and adventitia or serosa. We looked at the histological layers of the esophageal wall, the mucosa, submucosa, the muscularis, and adventitia or serosa. Now, the second part, which is the longer part of the lecture, is going to be about the stomach. So, histologically, the stomach is divided into 3 regions. The cardiac region, which is near the esophagus, the pyloric, which is near the sphincter. I'm sure Dr. Akif talked about that also, and the fundus, which is between the 2 regions. I think anatomically it's divided into four. I think there's another fourth area. I don't remember what it is. But histologically, they are actually, the majority is the fundus. Most of the stomach is basically the fundus here. It's called fundus, but actually, I think in the anatomy, they call it fundus in body. But here for us, fundus is everything, and histologically. Here is the cardius, very small area, and the pyloric, everything is fundus. And I'm sure also Dr. Raik covered this. This mucosa is forming those prominent rougets, these folds when the stomach is empty. They have all these wrinkles made of the mucosa. They actually help when the stomach stretch. So you have actually some room for the stomach to stretch when you fill your stomach with food. Now again, we'll cover the major four layers. The mucosa, which again thrown into these prominent folds. It contains the gastric glands that extend from the level of the muscularis mucosa to the open. Again, this we'll talk about all this in details. It's just a general structure. So we have mucosa, submucosa, and then we have the layers of the muscle. The submucosa, again, relatively loose here and extensible, and they contain large blood vessels. The muscularis actually here has a third layer. We have always outer longitudinal, inner circular, but this hand actually have an inner oblique. Again, the differences between this on sections is difficult. Sometimes you cannot actually tell which one is the oblique, but there is a third layer. It's also not as developed or well developed as the other two layers, but it is there. The fourth is the serosa because it has a peritoneal covering from the peritoneum, so we call it serosa, not adventitia. Now, here is the actually important part coming, which is the gastric gland. This is probably the key slide here for today's lecture. So the fundic glands, they also call it gastric glands, present throughout the entire gastric mucosa, except for regions occupied by the cardiac and pyloric glands. So this is the surface of the stomach. You can see this invagination. You can see this hole inside. It's going deep through the mucosa. So you can see this ends up here, and you can see all these cells. Again, they look like gland. They're actually tubular gland. So this, we call it the gastric gland. So this is not elevation. It's not like the papillae. It's not going up top of the surface. Actually, invagination. It is a drop inside the surface. So this is called the pyloric, the gastric gland. There are simple branched tubular glands extend from the bottom of the gastric pit. So the whole thing is not the gland. The invagination that starts, they call it gastric pit. Pit is a hole. So, and after that, there's actually two or three glands connected to it. So if you remember the diagram of the tubular gland, you have something in common, like a common tube, and then the branching. It's the same here, but the upper part, we call it the pit, which is a little bit long, and then you have the branching of the gland coming. They extend from the gastric pit, which here, all the way to the muscularis mucosa. So basically, if you look at the gland and the pit, it basically cut across the whole mucosa, starting from the very top surface all the way to the muscularis mucosa. So it's all occupied by either pit or the gastric gland. This is a nice image using scanning electron microscopy. You know, the scanning electron microscopy basically give you an image, detailed image of the surface. So we're looking inside the stomach at the surface. So these are cells, mucous cells we're going to talk about. They look like tiles, correct? And these are the pits, holes. So if you have the surface, this would be the pits here, like where you can see the electric, this would be a hole, this would be the pit. So this is going to be the tiles, the surface of the stomach, and these are the pits. But they go deep. These not as, I mean, these are shallow. So this is an illustration matching what the pit and the gastric gland looks like. So here is the surface of the stomach. This is the pit. The lumen is the opening. This is the pit, is the hole. And then you have the glands, several glands connecting and, draining the secretion inside the pit. So the glands will be doing something here, and all the secretion is going to come to the surface of the stomach. So if you look at the stomach, sometimes you can actually see there with the naked eye. It looks like a pinholes. Somebody punched inside. With the scanning electron microscopy, so this hole here, is this one here and it's this one here. If you look deep inside this hole, this is what you're going to find with the histology. Okay. The main thing here I want to also point out is something called the isthmus, which is this part. It's actually, you need to remember that because it's difficult to see that in the histologically. And again, you see, it's difficult here to define where the beginning and the end. I mean, this histology, things are going in different directions, and so as the glands. But if you look at the illustration, which is the perfect, position, this area called the isthmus, containing the stem cells. We have a continuous renewal of the cells. I mean, you lose these cells all the time. Some of this only a few days, especially the surface. So you have a very active stem cells at this area, and the cells go either up or down based on the need. And they can differentiate to any of the other cell types we're going to talk about. So when I said gastric glands, because the glands are a little bit different between the regions. The main one between, as I said, the gastric or the fungus of the mostly majority of the body of the stomach. The other one actually region has a substantial area, it's called the pyloric area. So the difference is that the pyloric glands, the gastric pits are deeper. If you look at this one, you can see actually they're going really deep compared to the rest of the gland. So the pit itself is deep compared to the funding. They don't go as deep. And the predominant type of epithelium, and they also have larger lumen. If you look, sometimes actually better images of this. The predominant epithelial type is a mucus secreting cell that resembles the mucus-like cells of the gastric glands. The cells secrete mucus and granules containing lysozyme. The cells are a little bit different than the rest of the gastric glands. We'll talk about that in details in the next few slides. So if you look at the gastric glands, there are 5 types of cells present there, not only one. The first ones, we call it the mucus cells. These are basically the surface cells that's covering the surface of the stomach. And the mucus neck cells, which actually, it goes a little bit deeper inside the neck here. There's also cells called chief cells. Sometimes they call them the peptic cells. The parietal cells, called sometimes oxyntic, but the chief cells and parietal cells is the term we're going to use. There's also the enteroendocrine cells. They used to call them in some of the old books enterochromaffin, but now they actually find the function. Before, it was actually because of the color. This chromaffin was actually specially stained. They can pick up those particular cells. They are scattered among the other cells, and then we have stem cells that I told you will be present at the isthmus. So the first is the gastric mucus cells. Again, when you hit the word mucus, it means the same thing. It's filled with mucus. So they line the surface and the gastric pits. So if you look at the surface of the stomach, they're all lined with these mucus cells. And also this is then going to be the pit, which is the hole, always covered by these mucus cells. The lifespan is short. In 3-5 days, you lose those cells, and then the stem cells will be replacing these cells. They are simple columnar, again, like a lot of secretory cells, and have the apical mucus cup because of the mucus, again, pushed to the apex of the cell. The mucus is high in bicarbonate and potassium because to protect from the acidic secretion. We'll talk later that the stomach makes acid, and you need actually to protect these cells, the neighboring cells from the acid. And that's why they have this mucus, it's thick, and it's high in carbonate and potassium to neutralize the acid. So protect the epithelium from the stomach acid. They secrete prostaglandins, which stimulate the secretion of the bicarbonate and increase the thickness of the mucus. So there are 2 factors here. The bicarbonate that's made, the prostaglandin made, stimulate the secretion of bicarbonate to make the mucus thick and also to neutralize the acidity of the stomach. I put this here as just clinical because it's very related. Do you know the word NSAIDs? This stands for non-steroid anti-inflammatory drugs. It's one of the most common things you will ever hear in pharmacology. This is aspirin, basically. Advil, this kind of over-the-counter thing everybody takes. It has a really bad effect on the stomach if people don't pay attention to this. So a lot of disease actually happen because of us taking this, without really paying any attention to the danger. It suppresses the production of prostaglandin. So this one thing, you lose the ability to protect your stomach from these drugs. And then you actually can injure your stomach, the mucosa, because of the acid, because they have nothing to neutralize. They are very, very potent. Yes, they're very effective, strong anti-inflammatory, but look at the side effects. That's why they told you always drink it with water, take it with water. after meal, that kind of thing. Still, the effect is really is there. And it's also the direct contact with the wall of the stomach interfere with the hydrophobic properties of the gastric mucosa. The gastric mucosa have the ability not to be actually hate water or whatever, repel water. But when you use these drugs, it actually cut this. So it actually increases the chances of the stomach being broken by the aspirin. The other actually cells, it's another mucus cells, but it's actually called the mucus neck cells. They're a little bit different. Again, this is an illustration showing the mucus cells with the different types. You know, they give them different color in here. So the mucus neck cells are those red ones. They are mostly present in clusters and they next to other cells as well in the gastric gland. They are less columnar than the surface mucus cells. So that's why they can identify them. So they're a little bit shorter. They are low columnar or lower columnar in this case. And the mucus secretion is less alkaline than that of the surface epithelium cell. So the surface epithelium cell are the one that's making actually the protection. Now the second cell is the parietal cells. This is predominate near the neck and the upper segment of the gastric gland. You can see them here. These are the parietal cells near the neck. Histologically, these are large, sometimes binucleate. You can see those cells. You'll see them in histological section like that. They're actually pale, rounded, larger than any other cell. Sometimes you can see the binucleation. I don't see it here. And sometimes they appear triangular, like this one looks a little bit triangular. They have abundant mitochondria, which provide the ATP to pump the proton, the hydrogen ions into the lumen of the secretory granules. Secretory or intracellular canaliculi. You have canaliculi for the secretions here. Then you can see that in electron microscopy or in illustrations like this one. There is an hydrogen potassium independent ATPase rich tubular vesicular system which kicks these ions out. The lifespan is long compared to the mucus. It's 150 to 200 days. So most of the time, those stem cells will be replacing mucus cells, not parietal cells. So the function of this acid. This is the cell that make actually the stomach acid, HCl, hydrochloric acid. The concentration is about 1 to 2. The pH is very, very low acidic. But these are cells that make your acid for the stomach. Initiate digestion of special dietary proteins. So you need the acid to break the protein. It also converts the pepsinogen into active pepsin. Pepsinogen is made by the chief cells. We'll talk about that. And this actually, the active compound is the pepsin. The acid will convert the pepsinogen into pepsin. Bacteriostatic. So, I mean, we eat a lot of stuff actually carrying bacteria all the time. And it's actually because of the acidity of the stomach, we get protection, we don't get sick from what we eat. So the acid is important. Don't say that I'm taking something to inhibit the acidity of my stomach because I can injure none. But you need that. But some bacteria can adapt to the low pH. H. pylori, a lot of people probably hear about that. The stomach bug, or whatever they call it. Helicobacter pylori. They have this contain large amounts of ureas that have to hydrolyze the urea, so create protective basic ammonia cloud. So the acid doesn't kill them. That's why they live in the stomach, despite the acidity. So when you give the treatment for this, they give them antibiotics, but also they give them, drugs to inhibit the gastric acidity. So this is a big function in the stomach. The parietal cells are really important. The other things also make those intrinsic factors. A glycoprotein binds to the vitamin B12, so essential for the absorption. If you have an issue with this, you get actually vitamin B12 deficiency. So briefly, how the acid is produced, I'm sure you're going to be covering this more details in the physiology. But the water inside the cell will split into the hydrogen and the hydroxide group here, as you see. And the hydrogen will be pumped into the gastric lumen through this HK pump, the hydrogen potassium pump. And while the hydroxide group will bind to the carboxyl dioxide group to form the bicarbonate here. The bicarbonate will be in exchange with the circulation of the chloride. So the bicarbonate will be out, the chloride will come inside the cell, and then the chloride will be out again in the parietal cell. So you have the hydrogen otherwise, so the HCl will be formed. So again, just a clinical note here. There is these drugs, proton pump inhibitors. If you hear about them, Prilosec, Nexium, I think you see probably on TV all the time. It inhibits this pump, so you never actually develop the acid. That's a good thing about this drug. Very potent. It's not neutralizing. The other drug used to neutralize the acid. This one doesn't, prevent the acid from forming by inhibiting the adrenaline going outside. So it's a very potent drug. Again, there is little side effects known so far, but it's very, very powerful for many diseases. The other cells here we're looking at are the chief cells. Again, they are predominant in the lower third of the gastric gland, not present in the cardiac area, and seldom in the pyloric. So usually the major part of the body. They have abundant rough endoplasmic reticulum in the basocytoplasm, giving the cell this basophilic appearance. So when you look at them under the microscope, you can see this darker lower end here. That's why you can recognize these cells. They contain zymogen granules in the apical region, so they secrete this. The lifespan is 60 to 90 days, so they're somewhere between the mucous cells and the parietal cells. It's just an example with the illustration showing where the zymogen granules are. Again, as I mentioned earlier, the main thing they do, they make this pepsinogen, convert it to pepsin by the HCl at a lower pH of 5. So the parietal cells will make the HCl, the chief cells will make pepsinogen, and then pepsinogen will be converted into pepsin by the HCl. It hydrolyzes proteins into small peptides, so actually have absorption and later. And these peptides are further digested into amino acids by the enzymes in the intestine. So they break it for the intestine, so it's ready to be digested. Enteroendocrine cells. Again, they used to have a different name, but then enteroendocrine, they used to call them the neuroendocrine cells. But I think it's not much acceptable, although some people use it because it doesn't have much relation to the neurology or neural origin. But actually, they are endocrine organs. So this is a new thing, and in the future, you'll learn more about this. I think it's a lot of development in this area. They cause a lot of disease. They do more than what we thought they do. There are different types. It's not one type. They group them into this enteroendocrine, but there are multiple types to secrete different things. And every time we find something new about them, causing diseases, tumors arise from them, or do something to control the body homeostasis. They said this is actually the biggest endocrine organ. Although they are not really an organ, scattered everywhere, but actually, if you put them together, they'll be the biggest endocrine organ. There are various cells secreting various peptides, as I said, serotonin, gastrin, so on. The lifespan about 60 to 90 days. Peptide hormones have the following. These are general function. What can they do? Again, we cannot, if you look at the textbook, you find a couple of tables with all kind of secretions, peptides they make, and the function. So this will be interesting to read, but I don't think you should, maybe physiologically it will be important that you have to memorize, but for us, I don't want you to know the details. I just want to know what these cells are. So the functional regulation of water electrolyte metabolism, regulate gastrointestinal motility, stimulation and release of other peptides, a lot of variable functions. Sometimes you can identify them in the sections. At the lower one, it's not reaching the base, but it's difficult. So I don't think it will be something that you need to spot on the exam. Types of endocrine cells, they divide them into two types, closed and cell and open. Basically, because if they reach the surface or not, similar to the pseudostratified epithelium, but reach. So the closed one is actually, you can see, does not reach the apex of the cell, does not reach the lumen. So they call it closed. The open one, actually, where the apex of the cell can reach the lumen and become in the surface with other cells. But functionally, I don't think there's a difference between them, at least when it comes to this description. But based on the other function physiologically, yes, there is. Stem cells, again, this is the cells at the isthmus. We said this is the one that actually generate and replace the cells found at the isthmus. They produce progenitor cells for all other epithelial types. So they replace the mucous cells, replace parietal cells, chief cells, anything. Some of these move upward to replace the surface mucous cells, which has a turnover, we said, in 3 to 7 days. So they probably will go up most of the time because these are cells that actually need replacement more frequent. Parietal cells, 150 days, the same. Chief cells, like 60, 90 days. So, and they are in the lower bottom. So you'll find most of this traffic is going to go up. not really down. So other cells, as I said, migrate deeply to differentiate into the secretory cells of the gland and so on. I put just this image because I really like it. It's actually showing a mitotic division of the stem cell at the isthmus here. It's difficult to spot, but you can see this actually the gastric mucosa cells. This is actually, you see that the isthmus here. This arrow is magnified. You can see the cell division. Very nice, actually. I just put it because I like it. It's not, and actually, I tried to zoom it in, it became fuzzy. But she show you actually how this cell division happened, and actually you can see it on the microscope. Usually in the late stages, not early one, but it's visible. And you know, an area like that, if you look carefully under the microscope in these slides, you'll find several of those, and you know that here's the isthmus. Otherwise, difficult to find the actual location. So we finished with all this about the mucosa. So this is really the main bulk of the lectures about the mucosa of the stomach. The glands is very important. So after that's lamina propria. So the lamina propria scant surrounding the gastric pits and glands, because the gastric glands pits occupy the whole mucosa. There's not really much until the muscularis mucosa. So there's not really much room for lamina propria, just supporting all the pits and glands. It's just filling the spaces between them. Again, stroma composed largely of reticular fibers, some fibroblasts, again, immune cells, lymphocytes, plasma cells, macrophages, the normal. So this is the lamina propria. The muscularis mucosa, again, has a smooth muscle layer. This is the last layer of the mucosa. It helps the gastric secretion also pump out. The submucosa, so we have the mucosa, submucosa composed with this dense connective tissue. Fat cells also can be present, as you can see, blood vessels. The submucosal plexus or mesenteric plexus similar to the esophagus. The mesenteric plexus present in the submucosa. The Auerbach's plexus present in the muscularis. The muscularis, again, 3 poorly defined layers of the smooth muscle. Again, as I told you, you should find 3, but a lot of times it's difficult to find oblique. So, but theoretically they are there. It depends on the section where you actually can see them or not. And you can find this bundle of unmyelinated nerve fibers, again, in ganglion present between the 2 layers, between the outer longitudinal, inner circular. These are the myenteric. You can see these are nerve cells. You can see, again, depends on the cut. You can see actually a bigger area. You can see full ganglion is there. At the end of the muscle layer close to the pyloric area, we have the pyloric sphincter, which is at the level of the distal pyloric antrum. These are circular muscular layer thickened. The muscle layer became thick here to form the sphincter, so to control. This is, you know, the importance of this is to prevent regurgitation. So when it will open only when you eat, but to prevent the food, going to the other end of the intestine. The serosa, which is the continuous with the partial parietal peritoneum, again because of the stomach, so it's serosa here, not the tissue we call it. Now I mentioned about the myenteric or enteric nervous system, which is the mesenteric plexus and the Auerbach plexus. It is considered the 3rd division of the autonomic nervous system, so it's not either one. They're responsible for innervating the smooth muscle of the alimentary tract. You find this only in this enteric, it means the intestine. So it's only present, it's an autonomic present only in the intestinal tract, some of the small stomach intestine, large intestine, small intestine. Function totally independent of the central nervous system. Present in the submucosa, constitute the submucosal plexus, or mesenteric plexus, on the muscularis, between the 2 layers, the myenteric plexus or Auerbach plexus. Just different name because of their location. You're containing nerve bodies, you see that on the microscope, ganglion, postganglionic, parasympathetic neurons, as well as neurons of the enteric nervous system, so some mixed. Now, this is going back to the area between the esophagus and stomach. We have the mucosa and the epithelium covering the esophagus as stratified squamous epithelium, protected. But in the stomach, we found these mucus secreting cells. The transition here, we call it the epithelial transition zone or the gastroesophageal junction. This is impressively precise. This is a histology of this area. You can see it's 1 cell basically difference. There's abrupt change. All of a sudden, from stratified squamous all of a sudden change to the mucus secreting. Very impressive how these things, there's no gradual changes. Boom, one disappeared, the other one stopped. Clinically, this is very important. The gastroesophageal junction is a transition from the stratified squamous to the simple mucus secreting of the stomach. This area can be identified under a microscope. Actually, with the endoscopy, if you look with the stomach, you can see actually a line. And I'm going to show you some images. And changes here suggest a disease process. We'll talk about that in the next couple of slides. This clinically, this is probably the most important thing you need to know today. You have to remember the gastroesophageal, sometimes we call it esophageal gastric, but it's always known as a gastroesophageal. What's the difference here? The squamous, stratified squamous will change into simple columnar, which is the mucus secreting. It's a abrupt transition. This defines where the esophagus ends and where the stomach starts. So the disease of the stomach, the GERD, the gastroesophageal reflux diseases, I think if you work in any specialty, you will find somebody with this condition. Very, very common thing, with a variable degrees. I have this disease since I was 12. I have this reflux. So I know exactly what it is. So it occurs when the lower esophageal sphincter is weak or relaxed. So normally, the sphincter at the end of the esophagus will open to pass the food, and when the food is passing in and swallowing, it will close. So none of the stomach contents will kick out back to the esophagus. But if it's not closing properly, it's going to push some of the food out of the esophagus. Very painful. Turn actually the stomach into something, in the esophagus into something. The stomach contents will flow into the esophagus, causing a burn sensation. Call it pyrosis, heartburn. It's not ear burn. It's not a spelling mistake here. Heartburn. A lot of people describe it. I have burning in my heart. I mean, this is what they point at. But when they said that, usually it is this thing. See here, this white thing is endoscopy. You look inside from the esophagus down to the stomach. The white thing is the stratified squamous epithelium of the esophagus, and this red is the stomach. It should be very well demarcated line like this one here. So you can see here what happened. It's actually changing. Now the epithelium of the esophagus, the stratified squamous, is replaced by columnar. Again, the columnar is actually red because it's thinner, so you can see the blood vessel underneath that, so it looks red. But when this changes, we call it Barrett's esophagus from the GERD. GERD is just going to be simple some inflammation, but when actually the epithelium change, metaplasia, I don't want to give you terminology which is related to pathology, confuse you, but just understand this. The replacement of the esophageal squamous epithelium by columnar. Because what the body was trying to do, I mean, this acid is burning in the esophagus. We have to make it to another epithelium that can tolerate this. And the mucus secreting cells of the stomach is protected, So turn into this. The problem with this, it can turn into malignancy after some time. I mean, the chance is not high. I think like 1% of the people with long GERD, 1% of them will develop Barrett's esophagus, and 1% of the Barrett's will develop adenocarcinoma. But this is a common thing. The people is, when they ignore the treatment of this, if they have really changes, it's dangerous. So you have to take action. And it's not a simple thing. And again, because of just how common this is, I mean, it can get worse. The other thing is also helpful in make this worsen is the food we eat. The caffeine is a terrible thing. Smoking, alcohol, all the stuff make things worse for those people. Chocolate, I used to eat chocolate like crazy, all the time. In the US, the chocolates are unbelievable. But I have to stop because of this. Because of the caffeine contents. It's very high caffeine. It's worse than the coffee and the tea. Of course, it's things like soda. But what I'm talking is a lifestyle modification can make a difference. That's why a lot of time you need actually, I'm saying that because a lot of time you find your family members asking you, so at least you can tell them something. Lifestyle modification will help a lot of things, hypertension, diabetes, GERD. So it's a good thing that I know things can get actually worse. So please do this, this, and this. You don't have to be a doctor, or certified doctor yet. But definitely things like it's not going to hurt them, it will help them. But this one of the things can be preventable to get better, but a lot of people don't pay attention, just keep doing the same thing. But again, this one thing, you can see histology actually in action. No, the histology help. I know this stratified squamous epithelium is not stratified squamous epithelium anymore. It's changing into something different, and it should. Yeah, why? It's trying to do a protection, but in the meantime, also it's a problem. Esophageal cancer, it might rise again on top of the varus esophagus, because this actually can happen. And this lesion here in the esophagus, I mean, they can treat it to some extent. It depends on the stage of the cancer. But you can see this actually can develop from here to here. Again, it's not a common thing, but actually it's there. Other disease of the stomach, Zollinger-Ellison syndrome, somebody's name, 2 people actually. So it's not. It's a gastrinoma. It's a tumor, actually a neuroendocrine tumor. I told you there are different types. This is the one that's making gastrin, and gastrin stimulates HCl. So it's excess secretion of gastrin by a gastrinoma of the duodenum or the pancreas. So what happened? Excessive secretion of the hydrochloric acid because of the continuous stimulation. The gastrin stimulates the HCl, so parietal cells keep going. So even with the presence of those protective secretions, but when you have overwhelming amount of acid, then you will develop an ulcer. So they erode, the acid at the end will erode the protective mucosa because it can keep up with the secretions. So you end up with these ulcers. So the excess acid cannot be adequately neutralized by the stomach. You know, you just keep adding acid all the time, leading to this gastric or duodenal ulcer. As you can see in this image here, you can see the gastric mucosa almost gone. Again, proton pump inhibitors, Nexium, and some. This is also really changing the game here. When you know that Zollinger-Ellison was difficult, you have to remove the tumor. There's no other, because anything else you use, Tums and stuff, it doesn't help. You produce acid like crazy. The proton pump inhibitor actually give them, at least a chance that the tumor is removed, or even if they stay, they can neutralize. It's very, very potent. Again, it inhibits this hydrogen potassium pump, so there is no hydrogen coming out. You cannot make the HCl. So for those people, actually, it was a big change in the game here. To summarize this part, we talked about the histological layers of the gastric wall, mucosa, submucosa, muscularis serosa, structure of the gastric pits glands. We talked about the various types of the gastric glands. We talked a little bit about the HCl production by the parietal cells. I just want you to know the main parts. This actually came, how it came, where the acid came from, how where the chloride came from, where the hydrogen came from, and some disease of the esophagus and stomach. We're not going to test you on the disease. I put it here just to link. I might put it in the question to link you back to the cause, to the tissue changes, but not to ask you about the disease. So it's going to be a distraction if you put the disease and asking about the disease. So don't think about it. Think about what the disease means in histological terms. Okay? I'm not giving you a clue, but I'm giving you what I want you to know. Thank you. Attendance. I almost forgot. I'm not used to this.